Hello everyone. Today's topic is pharyngeal arches. Pharyngeal arches is the most important topic of the head and neck embryology. First of all, the definition of the pharyngeal arches. How we define the pharyngeal arches. Pharyngeal arches consist of the core of the mesenchymal tissue. It consists of the core of mesenchymal tissue, which are covered on outside by ectoderm and on inside by the epithelium, which is derived from the endoderm. So these pharyngeal arches are seen during the 4th and 5th week of development. During 3rd week embryo, 3rd week is known for the gastrulation. Gastrulation means 3 germ layers are formed. In 4th week we have pharyngeal arches in the cranial portions at the neck region, future neck region of the embryo. This so ऐसे याद रख सकते हैं 3 लेयर्स हैं. बाहर की तरफ एक्टोडर्म, अंदर एंडोडर्म डिराइव्ड एपिथेलियम और बीच में मेसेनकाइमल और कोर ऑफ द मेसेनकाइमल टिश्यू. Now, this, if we cut the embryo through this, the cut section, please find this. In 4th and 5th week of the development, initially क्या होगा? We have bar of mesenchymal tissue. We have bar of mesenchymal tissue, which is separated by deep clefts on outside, and these clefts, ectodermal clefts, externally we have these deep clefts, and these clefts are termed as pharyngeal clefts. Simultaneously, externally we have clefts, internally or from inside we see the out pocketings or pouches appear along the lateral wall of the pharynx. See this diagram. This is the future mouth or stomodium. This inner lining is formed by the epithelium, which is derived from the endoderm, and this is the future. This is the stomodium, that is the future mouth, and below the stomodium we have, in continuation with the stomodium, we have the cranial most portion of the foregut that develop into the future pharynx. And from the lateral wall of the pharynx, if this is the pharynx, from the lateral wall of the pharynx we have out pocketings or pouches are formed, and these pouches penetrate in the surrounding mesenchyme. तो क्या है कि बाहर की तरफ हमारे पास एक्टोडर्म है, बीच में मेसोडर्म है अगर हम 3 जर्म लेयर्स की बात करें, और अंदर की तरफ ये pharynx इस एंडोडर्म है. तो एक्टोडर्म पेनिट्रेट करेगा मेसेनकाइम के अंदर 4th and 5th वीक के अंदर और फॉर्म करेगा pharyngeal clefts. एंडोडर्म, लाइनिंग ऑफ द एंडोडर्म, ये दैट इज फ्यूचर pharynx की जो लेटरल वॉल है वहाँ से पॉकेट्स बनेंगी इस तरीके से, और ये पॉकेट्स भी पेनिट्रेट करेंगी किसमें? सराउंडिंग मेसेनकाइम में, बाहर से अंदर की तरफ और अंदर से बाहर की तरफ. बाहर से अंदर जो आ रहा है दैट इज द क्लेफ्ट, और अंदर से जो आउट पॉकेटिंग्स बन रही है एंडोडर्म की और फ्यूचर pharynx की लेटरल वॉल से मेसेनकाइम के अंदर, वो फॉर्म करेंगे pharyngeal pouches. बट इन ह्यूमंस में क्या होगा? ह्यूमन एम्ब्रियो के अंदर pharyngeal pouches pharyngeal clefts के साथ मिलेंगे नहीं, कभी भी डायरेक्ट ओपन कम्युनिकेशन नहीं होगा. ओपन कम्युनिकेशन सीन कहाँ दिखाई देता है? फिशेस में और एम्फीबियंस में. फिश के अंदर गिल स्लिट्स बनते हैं, ह्यूमन के अंदर गिल स्लिट्स नहीं बनेंगे, बस वो प्रोसेस जो है वो बीच में रुक जाएगा. वहाँ पे फेरेंजियल मेम्ब्रेन्स बनेगी, जहाँ क्लेफ्ट और पाउच, एक्टोडर्म और एंडोडर्म बिल्कुल पास पास आ जाएंगे। तो टर्म है फेरेंजियल apparatus. Pharyngeal apparatus is formed by pharyngeal arches, pharyngeal cleft, pharyngeal pouch and pharyngeal membrane. If we see in this diagram, this whole portion, this shaded portion forms the pharyngeal apparatus. Pharyngeal apparatus में ectoderm भी आ गया, that forms the cleft, mesenchyme आ गया, that forms the arch, and endodermal lining आ गई, that forms the pouches, और ये वाला grooves, ये वाला जो पोर्शन है ना, ये जहाँ पे एक्टोडर्म और एंडोडर्म बिल्कुल पास पास में आ जाएंगे, that portion is termed as pharyngeal membrane. Only the first pharyngeal membrane persist in humans, and that forms the tympanic membrane. Our form appear during the 4th and 5th week of embryonic development. Total pharyngeal arches formed in humans are 6 in number, but fifth arch disappears. So remaining arches are arch 1, 2, 3, 4 and 6. Now each arch consists of skeletal component. Each arch consists of its own skeletal component, its muscular component, its neural component and its atrial component. Arch is formed by the mesenchymal tissue. Now how this mesenchymal tissue get converted into the skeletal component, muscular, neural and atrial? Now, each arch have mesenchymal tissue. Plus neural crest cells. These neural crest cells arise from the neuroepithelium and neuroepithelium of the neural folds. After formation of the neural folds under the influence of the bone morphogenic protein, that is the BMP, under the influence of the WNT1, that is the Wnt1 gene, these neural crest cells migrate to the pharyngeal arch region and they undergo epithelial. These arise from the neuroepithelium, but they get converted, they have the potency to change into the mesenchymal tissue. So under the expression of the WNT1 gene, these neural crest cells migrate and they undergo epithelial to mesenchymal transition. And these neural crest cells are very important for the development of the skeletal components of the pharyngeal arches. How? Let's see. In the developing hindbrain during the 4th and 5th weeks, as we see in this diagram, this is the developing hindbrain region of embryo in 4th and 5th week. These hindbrain crest cells, the neural crest cells of the hindbrain originate in a specific pattern. They form these segments. These segments are termed as the rhombomeres. These segments are termed as rhombomeres, and these are 8 segments, R1 to R8. Neural crest cells from the specific segment of the hindbrain, that is the rhombomere. From specific rhombomere, neural crest cell migrate to the specific arch, and this is very important. And this neural crest cell migrate in rhombomere, neural crest cell migrate into the specific arch. And neural crest cells migrate in 3 streams. As we see in this diagram. This is the hindbrain region. These are the 8 rhombomeres, and from R1 and R2, that is the first and second rhombomere, and from the caudal region of the midbrain, neural crest cell migrate into the first pharyngeal arch, and from the 4th rhombomere, the neural crest cell migrates into the second pharyngeal arch, and from the 6th and 7th rhombomere, from 6 it migrates into the pharyngeal arch 3, 4 and 6. From 6 and 7, and the specific neural crest cells undergo specific epithelial mesenchymal transition to form skeletal component of particular arch. So these rhombomeres migrate into the these pharyngeal arches and form the skeletal component. This migration is also important because these three streams provides migration. These migration paths provide axonal guidance for axons from the ganglia. Now, neural crest cells undergo the epithelial mesenchymal transition. Neural crest cells undergo epithelial mesenchymal transition under the influence of Wnt1 gene. तो जितने भी न्यूरल क्रेस्ट सेल माइग्रेट होके आए सबके सब कन्वर्ट हो गए मेसेनकाइम के अंदर, पर्टिकुलर आर्च के अंदर. अब पर्टिकुलर आर्च के अंदर, स्पेसिफिक आर्च के अंदर जो मेसेनकाइम है वो स्केलेटल कंपोनेंट, मस्कुलर कंपोनेंट कैसे बनाएगी, कौन इन्फ्लुएंस करेगा? मेसेनकाइम फॉर्म इट्स डेरिवेटिव अंडर द इन्फ्लुएंस ऑफ skeletal component is formed by the neural crest cells, and it is formed under the influence of the endoderm of pharyngeal pouches. So it is very important to note that pharyngeal pouches are formed before the neural crest cell migration. Neural crest cell migrate after the formation of the pouches, and endoderm of the pharyngeal pouches constantly stimulate and give endodermal signals to neural crest cells to get converted into the skeletal component of the particular arch. So mesenchymal derivatives or mesenchyme of a particular arch responds on the endodermal signal from the pharyngeal pouches and from the transcription factors expressed in the particular arch. Transcription factors of the particular arch, that is the Hox genes. Hox genes are homeobox genes. Now, pharyngeal arches, component of the pharyngeal arches. The pharyngeal arches appear in 4th to 5th week of the development, 6 in number, 5th arch disappear. Each arch consists of the skeletal, muscular, neural and atrial component. Now skeletal component derived from the migrated neural crest cells. In the original mesoderm of the arch, original mesoderm or the mesenchyme of the arch form the muscular component of the arch, muscular component, also termed as the bronchomia. Muscular component migrate and carry their nerve supply with them. So it is not necessary that the muscle develop from the particular arch. For example, muscular components develop from the first arch is attached to the skeleton component of the first arch. It is not necessary. Muscles can migrate, get attached to another bony structure or another bone which is not derived from that arch. Then how we identify that particular muscle is derived from that arch? This is confirmed by the nerve supply of that muscle, because muscular component migrate and they carry their own nerve supply. And for the atrial component,